Good afternoon. Can you hear me in the back? Okay. I am Prem Paul, Vice Chancellor for Research and Economic Development, and it is a pleasure for me to welcome you and thank you for joining us today for our Fall Nebraska Lecture, the second of the 2008 Chancellor's Distinguished Lecture Series. The Nebraska Lectures is an interdisciplinary lecture series designed to foster communications among students and faculty in different academic areas and the people of Lincoln and Nebraska. These lectures are sponsored by the UNL Research Council in cooperation with the Office of the Chancellor and Office of Research. The Research Council is composed of faculty from across all disciplines at UNL. The Council sports several competitive awards and programs designed to encourage and enhance research, creative, and scholarly activities for all UNL faculty. The Nebraska Lectures is one such great program. The Research Council selects Nebraska Lectures from UNL faculty on the basis of major research recent accomplishments and the lecturer's ability to explain their work to a multidisciplinary audience. Now, just a few words about today's format. Following Dr. Kamil's presentation, Dr. William Wagner, Associate Professor in the School of Biological Sciences and a Research Council member, will moderate question and answer session. After this discussion, please join us for a reception across the hall in the Regency Suite. I also want to introduce our two sign language interpreters for today's lecture, Kelly Bricknoff and Nancy Bird. Please join me in thanking them for their assistance. Normally, at this point in these Nebraska lectures, I introduce Chancellor and go and sit down quietly and listen. But today it is my pleasure to fill in for Chancellor Perlman. Dr. Kamil, Chancellor Perlman wishes he could be here and asked me to personally congratulate you and thank you. Today's speaker believes it is an honor to be called a bird brain. In fact, his research proves that point. Dr. Al Kamil has studied the way birds learn and use memory for more than 35 years. His research interest is how birds' cognitive abilities affect the evolutionary process. His work challenged the traditional notion that birds have limited intelligence. He has shown that birds have highly developed cognitive skills that help them remember the location of food, infer social status, and detect prey. Dr. Kamil is the George Holmes University Professor of Biological Sciences and Psychology and joined the University of Nebraska-Lincoln faculty in 1992. He is director of the School of Biological Sciences and was director of Cedar Point Biological Station from 1999 to 2005. In 2004, he received an Outstanding Research and Creativity Activity Award, or for short, ORCA, the highest research honor conferred on faculty by the University of Nebraska system. He holds degrees from Hofstra University and the University of Wisconsin, where he pursued interest in experimental psychology and animal learning and behavior. Before joining UNL, he was a psychology department faculty member at the University of Massachusetts in Amherst. When he came to UNL, he switched departments and joined the School of Biological Sciences. It's been a great fit for his research interests, which have been funded by the National Science Foundation and the National Institutes of Health. Dr. Kamil's early work on bird intelligence was all but ignored because it so contradicted accepted notions that birds are stupid. But he was undaunted because he knew he was on the right track. Dr. Kamil decided to tap his expertise as an experimental psychologist well trained in laboratory techniques to study questions known to be important in the field. He employed the rigor of these techniques in both the field and the lab. Since then, he and his colleagues have shown time and again that birds learn and adapt to their environment. and that has become the accepted view among his peers worldwide. In 2007, he was awarded the Quest Career Award by the Animal Behavior Society for his outstanding seminal contribution in animal behaviors. I remember that at the time that not only we were excited, but the funding agencies took great pride in highlighting that accomplishment. So perhaps calling him a bird brain is a high praise. His lecture today is titled Bird Brain, Compliment or Insult. So please join me in welcoming Dr. Al Kamil.
I want to make sure I'm on time, or at least close. I don't want Bill to have to come up and shut me up. So first of all, I'd like to thank Prem for that very generous introduction. Can you hear me all right in the back? Okay. I'd like to thank Prem for that generous introduction and thank the Research Council and the Vice Chancellor's Office and the Chancellor's Office for the honor of this invitation. I really appreciate the honor of being asked to deliver a Nebraska lecture. And I thank all of you for attending. I just hope that what I have to say lives up to expectations. Before I get started into the meat of the talk, so to speak, I want to say the talk is divided into 3 parts, each lasting about 15 to 18 minutes. The first part is kind of introductory, and it will lay the groundwork of some of the special problems that those of us who study animal intelligence or even animal behavior face in doing our research and explaining our research to people. And then we'll talk about some of the very early work that I did when I first started working with Corvids, members of the crow, jay, nutcracker, magpie family. And then I'm going to talk about 2 specific examples drawn from about half a dozen of work that we've done using the technique that Prem described so well, looking for situations where animals use cognition in natural settings, bringing those into the laboratory in most cases for careful experimental analysis. One case is one that I've worked on with Russ Balder of Northern Arizona University and a lot of other people since 1982. And I have a lot to say about that. I'll just hit a couple of highlights. And the other is work that I've been doing most closely with Alan Bond, but also with Russ Balder and a variety of other people here at UNL, looking at social cognition in pinyon jays. And we've only been working on that for 7 or 8 years, so we have less to say about that because this work is not fast. It's slow, difficult work. So that's kind of the broad outline. So let me start with the problem of folk psychology that many of us face. So there is a very strong desire, apparently, in the human mind to attribute human characteristics to animals. And this really makes it difficult for those of us who study animal behavior in general and animal intelligence in particular, because a lot of times people think we're studying the obvious. They know the answer. And after all, we've all got pets, we've all seen animals, and we all know what wonderfully intelligent things they can do. And often we're jumping to conclusions. We see an animal do something that looks like something that we do, and we assume that the animal is doing it with the same cognitive capacity, the same thoughts and feelings that we have. And that's just not necessarily true. There's a very famous case of this in the history of experimental psychology called Clever Hans, which many of you probably heard of. Hans was a horse, and he was quite clever. And he could answer any mathematical question you cared to give him, provided the answer was about 10 or less. Okay? And in fact, they appointed a panel of scientists, biologists, and physicists, and so on, to look into this. And they concluded that it was real, that there was no fakery, and that Hans had the mathematical ability of the average 14-year-old child. Okay? So I'm not making this up. Okay? But then an experimental psychologist named Otto Funks got involved, and he started studying it very carefully, and he noticed that when the distance between the horse and the person asking the question increased, the horse's error rate went up. And he got suspicious that the horse was doing something about what he could see in the questioner rather than the arithmetic. And it turns out that's exactly what Hans had done. Clever Hans was very clever indeed. He had learned to pick up on the body language and the facial expression, what poker players call the tells, okay, the little involuntary things that give away what's going on. And if you put up a screen so that Hans couldn't see anyone who knew the answer, not only could he not answer the question, but he got really annoyed. Okay. So, and this was apparently innocent, but it wasn't always innocent. And if you go to many animal shows these days, trained animal shows, you'll see animals who can do arithmetic. And I got something off YouTube here. And let me find it. Okay. And so here's a young lady, and she's trained this parrot to count. She's going to ask it to count to 8 by ringing the bell. Okay, and let's see if I can get this to start.
8 on your bell. No problem.
No problem. I love the way she says that. Well, the reason there's no problem is she trained the bird to ring the gong until she gave it a signal. And I meant to point that out before I started. When the bird walks up to the gong, her right hand disappears and goes under the table. Almost always what the signal is in these trained animal acts is like that, a light tap on the bottom of the table. So it may look like arithmetic, but it's not. It may be clever, but it's not arithmetic. It's not counting, okay? So does that mean that whenever we see a parrot and the claim is made that it can count, we should just assume that it can't actually count and it's a trick, intentional or otherwise? Well, if life were that simple, it would be nice, but it's not that simple. I'm absolutely convinced there's at least one parrot, he's now unfortunately dead, but at least one parrot that really could do something very similar to human counting. And that parrot is an African gray parrot that Irene Pepperberg spent many years working with. And and you may know this story because it's been featured on TV and in the news and so on many times. She had the really interesting idea, and it turned out to be brilliant, to teach the parrot, Alex, to use words but with reference. So he learned to name the colors of objects, he learned to name what the objects were made of, he learned to ask for things, he learned the words for numbers, and he learned to use them appropriately. And what I'm going to do now is I'm going to show you a video of Alex and Irene. Now this is, okay, I want to say a few things before we go into the video. Irene conducted these experiments under very rigorous conditions. And I visited her lab, and many other people visited her lab, and there's no question that there were no clever Hans effects involved or anything like that. But to make a demo tape, she didn't go into the lab. She's doing this in her office to give you an idea of the kind of thing that Alex was capable of. Okay, but this is not a clever Hans case. I think you'll see that from the nature of the way he answers a variety of questions about a particular array of stimuli he's probably never seen before. So she's holding up a tray. This is Alex, that's Irene. And on the tray, I don't know how clearly you can see it out there, there are 3 wool balls, there are 4, I forget, green or orange cubes, which she calls blocks. There are 6 orange or green ones. Alex will tell us, okay? And she's going to ask Alex how many, okay?
On the tray, how many orange wool? How many orange wool? 3. Very good boy. Okay, we'll get you that nut.
Note he asked for a nut. Do we know if he really wanted a nut? Well, he did. The reason I say that is because Irene did an experiment where every once in a while, when he asked for something, he would ask for many different things, they would give him something else other than what he asked for, and measured the rejection rate. When he gave them something other than what he asked for, he rejected it 80 or 90% of the time. When they gave him what he asked for, he rejected it only about 10% of the time. So I conclude from that he really did want a nut.
There you go.
See, I told you he wanted it.
How many orange block? How many orange block?
There are 6, which is a hard
orange
word.
Block? How many orange block? Very good. Okay,
the sibilants are hard for a parrot.
Green block. How many green block? Good boy. How many green block? Yeah, how many green block?
Never said it was easy.
How many green block? Good boy. How many green block? Now, how many green block? 4. Good boy.
So, if you see a parrot doing something that looks like counting, or any other animal for that matter, you're going to have to really investigate it to find out whether it's counting or isn't counting. The fact that it looks like counting isn't sufficient. So there's a lot of room for employment for scientists in this field. Okay. So I wanted to emphasize that problem because it frustrates many of us who work in this field, that people don't appreciate this problem. What I want to do now is turn my attention to the work we've been doing with Corvids, members of the J. Crow family. And we're very fortunate in Lincoln to have a couple of wonderful nature photographers who live in Lincoln. One of them is Joel Sartori, and he came to our lab last year and took a series of pictures. He's building up a library of images of different species. And I'm delighted to be able to show you these wonderful, beautiful pictures of some wonderful, beautiful birds. These are 4 of the Corvid species that I'm going to be talking about today. Now, I first got started with Corvids in 1968. Prem was generous when he said 35 years ago. And actually, first I started working with birds 5 years before that as an undergraduate. And I chose to work with the blue jay because it was a corvid that was available. I wanted a corvid that was good to work with. Why did I want to work on learning in corvids? Well, as Prem accurately described, the general view of the intelligence of birds was pretty dismal back then. Birds were generally thought to be stupid. And I thought that might be wrong. And the reason I thought it might be wrong were that the 2 pieces of evidence on which that opinion was based were each weak, in my opinion. One was based on evidence about the neuroanatomy of the bird. And there were 2 pieces of evidence. One is that birds, in general, have small brains relative to the size of their body compared to mammals. Okay. Secondly, when you look under the microscope at the structure of the bird's brain, there's very little cortex. And cortex is associated with higher cognitive functioning in mammals, especially in primates. So there were those 2 pieces of neuroanatomical evidence. The thing is, I knew that some families of birds had much larger brains than others. And corvids have very large brains. So, and the other group, by the way, are parrots. Okay. So I wanted to study, so I thought that was weak. And I also knew that although it's true there's no cortex in the bird brain, there is an area called Wulst and an area called hypostriatum, which appears to serve the same functions in the bird brain as cortex does. And so I thought the neuroanatomical evidence was iffy. There was also some behavioral evidence where birds really perform like they were really stupid in complicated tasks, but they were birds from families with very small brains, like pigeons and chickens. And so I didn't think that evidence was very good either. So I thought it was worth a try. Okay. And so we got a colony of blue jays going, and early on we got a completely unexpected indication that we might be on the right track. Tony Jones, who was a master's student working in my lab back in Amherst back in the late 60s, early 70s, walked in the lab one morning and he noticed that birds were doing this. They were taking pieces of paper, ripping them up off the bottom of their cages, putting it between the bars of their cage and using it as a rake to rake in food pellets. Tool making and tool using in birds. Okay, I went back and I found some 16mm film that we shot of this in 1972, and I had it digitized. So just for you, and for me later. Now let me find the right one. Okay, so here we are. It's slightly slowed up, so you can see it. They learned this on their own spontaneously, and it turned out almost every blue jay in our lab knew how to do it. Okay. And we did a couple of studies on it, and the first study that we did on it was published in Science, and it was a big fuss. You know, he got one. And so, and I thought it was important. Because it was one of the first really clear, well-studied cases of a learned tool use in a bird. And so I thought it was important for that reason, and I thought it was interesting, but people got really excited about it. And the question is why? And I think that part of the reason goes back to folk psychology. There's kind of an assumption that when you see an animal using a tool, it understands things about the physical world that we understand when we use tools. And actually, there's quite active research going on now with birds and tool use, as well as primates, trying to use tool use as a way to find out what they really do know about the physical world, not just assuming through folk psychology that it's similar to what we know. And Mario Pessendorfer, a new graduate student in our lab, actually has some blue jays in this situation now, and he may have some interesting things to tell us about it in the 6 months or so. We'll see. So counting and tool use are interesting, but they're interesting primarily because humans do them, not necessarily because they're important to animals. And I wasn't very satisfied with that framework, and so I started looking around for a different framework to find tests to test animals with. And one of the early tests that I tried was something called learning set. And I'm not going to go into great detail about this, but it's not necessary to the main point. It's a test where the higher this goes, the more intelligent, the better the learning is, okay? It's a measure of how well the animal learns to solve new problems it's never seen before. And you can see when you test a variety of mammalian species, they separate quite nicely on learning set. And these are all data from the 60s and before, okay? And not only that, but they correlate kind of with people's naive idea of evolution, where the closer they are to humans, the better they do. And so you saw this in all the textbooks. There was also one study with pigeons that was right beneath the rat, okay? So I thought, well, here's a task that's been shown to work with mammals. It was tested with a bird that I think is a pretty dumb bird. Let's try it with a bird I think might be a pretty smart bird. And so we did it with blue jays, and this is what we got, okay? And it's interesting, I wasn't going to mention this, but Prem did, so I'll reinforce it. What Prem said in his introduction, people ignored the work because it didn't fit. That's exactly what happened to this work. And we published, I don't remember how many, we were funded by NSF for 7 or 8 years for this work, published many papers on it, and you'd go to the textbooks and you'd see that birds can't learn learning set. It was quite infuriating. I learned something about the politics of science at that point. But this forced me to really confront the issue and ask the question, why? Okay, clearly you can't appeal to a closeness of relationship when the closest relative to the closest performance of the blue jays on this task is the rhesus monkey. The closest to the rhesus monkey is the blue jay. It's not relationship, it must be something else. And that's when I began to change from being primarily a psychologist, somewhat interested in biology, to where I am today, where I think of myself, well, I really think of myself as the line was drawn in the wrong place between psychology and biology for me. I'm right on the line, can't help it. It's where my interests are. But anyway, I started looking around for a different framework. And I got to thinking about the evolution of brain size, nothing terribly sophisticated or anything, but it hadn't been applied very much to cognition at that time. If you think about it, brain tissue on which cognition intelligence depends is expensive. It's particularly expensive to maintain. It's been estimated, I think a pretty good estimate, that the rate of energy use per gram per second in brain tissue is approximately equivalent to the rate of energy use per gram per second in the leg muscles of a marathoner running a marathon. And most of that is to pump the ions across the membrane to maintain the potential difference so the neuron can fire. Okay, well over 60% of it is for that purpose. So if you've got, let's say you have a bird out there or any animal out there and it's got a mutation, it's got an extra couple grams of brain in its head, it's paying a high cost for that brain. And if that brain doesn't give it something in return, better finding of food, avoiding of predators, attracting a mate, raising young, that animal carrying that extra brain is not going to do very well in the competition we call natural selection. And it's not going to leave as many offspring, and brain size will not evolve to be larger just because being smart is good. We tend to think that because we're human, and we depend on being smart. But there are many ways to make your way in the world with no brain at all. Okay? There are lots of animals. There are lots of animals and plants and all kinds of things that make their way in the world quite well. I don't believe there's anyone, I don't believe I've ever encountered a student. I've heard a lot of laughter from faculty on that. That wasn't my meaning. Okay. So this means, and this is a general principle, it's a good starting point for many research studies in evolutionary biology, that something that's highly costly must have adaptive function, or else it can't exist, because if it's highly costly, it's got to be giving something in return. And so I try to apply that hard. I wish it was original to me, but I think Darwin thought of it first. But I've tried to apply that in an area where it had not been systematically applied, and that's the study of animal cognition. And so it suggested to me an approach which was to go look for situations in the field the natural conditions under which animals live, where they use their cognition, or it looks like they use their cognition, and preferably, most of the time, bring it into the laboratory for study, although I have done some field work. But for the kinds of questions I want to look at, you really need to be able to control a lot of variables, and it's much easier to do that in the lab than in the field. And so we've concentrated in the laboratory. I also found out the hard way that there are lots, I could do field work, but there are lots of people who can do it as well or better than I, but I seem to have a particular something for lab work that makes me just, that's the better place for me to work. So anyway, so one of the cases that were studied is the Clark's Nutcracker, and the reason they're of interest is because they live in western North America at very high elevations, like these are the San Francisco peaks just north of Flagstaff, which is where Northern Arizona University is and where Russ Baldo lives, and at least until his recent retirement. And they live at those elevations year-round, okay? And, you know, it's pretty tough to live at tree line or near tree line in the middle of winter at, 9,000, 10,000 feet. There's no food, for one thing. There's no insects, there's no plant material to eat. How do they do that? Well, it turns out the way they do that is that in the fall, when the pine cone crop ripens, nutcrackers harvest pine cones, store them briefly in a sublingual pouch so they can fly around with 70 or 80 pine seeds at a time, and take them to areas where they store them. And they store them in lots of different places. And this is what it looks like. Pops it out of his pouch. I'm going to move it, put it back. Okay? And then during the winter, spring, and early summer, including the period of time when they're raising their young, they do this. Whoops. These are natural, these are actual unstaged wild animals doing it. Okay. So how do they find those seeds? Okay. Now, Russ Bald and I hooked up with each other, and he's an ornithologist, I'm an experimental psychologist, evolutionary biologist, hooked up at an ornithologist union meeting in 1981, I think it was. And in 1982, we submitted our first grant proposal, and in 1983, we started collecting data. And what the main question we wanted to hit first, I'm going to talk about the first experiment we did and then summarize a lot of our other results. We wanted to find out if the nutcracker really had separate memories for each cache site. Because the studies that have been done up to this point, where we started doing this work, what they did is they let birds cache in a room or an aviary, a dirt floor, then put the bird back in a couple weeks later and see if the bird could find its food at levels above chance. And the answer was yes, well above chance, even if you remove smell by taking some of the seeds out of the room. So that certainly suggests memory of some sort. But it could be, it's not necessarily memory for a particular cache site. It could just be some kind of rule that governs digging when you're caching and when you're recovering. So if a bird goes around with the rule of always dig on the north side of rocks and the south side of trees, and it goes around and does that when it's caching and when it's recovering, it's going to find its seeds much more often than you expect by chance, but all it has to remember is the rule, which would be very interesting. But is it really individual cache sites? So we thought the key was to figure out a way where we could make the bird cache in sites that we chose instead of sites that they chose. Okay? And then we sat around drinking beer on the floor of Russ's living room for several nights, talking about how blankly blank we could do that. And all of a sudden, one night, it must have been just the right amount of beer, but I can't remember how much it was. But anyway, we had, it was one of those things where one minute we didn't have a clue, and the next minute we knew how we were going to do it. And what we came up with was a room that looks like this. This is actually the original room in which we did the original studies in the 80s. And what we did is we built up a plywood floor, and we drilled out lots of holes, and we could put a paper cup in each hole. And the paper cup could either be filled with sand, in which case it was available to bury seeds in or to search for seeds in, or it could be filled like these over on the edge are, in this particular picture, with plaster of Paris or paper mache and painted gray. Okay, in which case it couldn't be dug in, and so it wasn't available. And so what we did was we put the birds in this room with most of the holes unavailable with paper mache cups in, and just a few holes open. Gave them a pile of seeds on the feeder. They were a little bit hungry. They came in. Nutcrackers will cache. I mean, you have to get them used to the room and so on, but they're born to cache. You give them pine seeds, they'll cache them, almost all of them, if you're just a little patient. And sure enough, they started to cache in the holes the few holes that we had left open. Then, 10 days later, and it doesn't really matter, it could be 10 days, it could be 2 months, it could be 6 months, 7 months, 8 months later, we put them back in here with all the holes open. If they can find their seeds then, we ruined any use of preference by just selecting a few holes using a table of random numbers. If they can still do it, then we've ruled out preference. And we've supported the idea that it's memory for separate cache sites. And here you can just see, this is the level of performance of each of 4 birds in that study. And you can see they do way better than chance, even when we select the holes. Okay, way better than chance. And this has been replicated using this technique and a lot of variants on this technique by us many times and by other people with nutcrackers, and other people have applied this technique to other species, including some mammalian species, and it works quite well with a wide variety of species. It's a really nice technique that we're really quite proud of. So, not only do nutcrackers do this well, I mean, that looks like pretty good memory, okay? And I mean, but how do you index it? Well, one way to index it is to compare it with other species. And what we've done is to use, again, kind of a classic biological approach to the problem. We selected species for comparison who are relatively closely related. They're all in the same family, but they differ in their dependence on stored food. Okay, so and we did this by taking animals, birds that live at different elevations in the Arizona area to begin with. And so we selected 3 species for the first study, nutcrackers, pinyon jays who live almost as high as nutcrackers, don't cache quite as much, so they're probably slightly less dependent on pine seeds than nutcrackers are, and scrub jays who live at much lower elevations. If you know Arizona, the ones we work with came from Kingman, which is high desert. It doesn't snow there very often. There's no time of year when they're completely dependent on stored food, but they do store food. And we tested them with exactly the same methods that I just showed you, okay? And we found that nutcrackers and pinyon jays were in a dead heat statistically, and scrub jays were still way above chance. Chance was about 0.06 in this experiment, as I recall, definitely below 0.1. And you can see scrub jays are well above chance, but significantly lower than pinyon jays and nutcrackers, and they also take longer to find their caches. They're slower at it. Okay. I have to peek now. Okay. So we've gone on, I mean, we've published these studies in 1985 and 1989, so you might imagine we've done a little work on this since then. And we're still working on some very interesting questions with nutcracker caching, although not as much as we used to. And I'm just going to summarize some of the things that we've learned about nutcrackers. Okay, they have their spatial memory is large capacity. The field data suggests thousands of sites are remembered, and we've never been able to get a room big enough to go that high. Although Northern Arizona University has an indoor football field and a bad football team, and I volunteered that. I thought overhead return might, pay better than football tickets at that particular university, not at this one. But anyway, it's very long term. We've tested them out to 285 days, and they're still well above chance after 285 days, although they've begun to forget a little bit. My guess is we never went back and did the study to really check out this hunch. My hunch is that they forget somewhere between about 280 days and 330 days. So they wipe the slate clean for the next year's caching. I suspect that, but I don't know it. We know that if you let them cache in a bunch of sites in a room and then you let them empty some of them, and then you put them in again later, they avoid the ones that they've emptied, so they either forget them when they empty them, but I don't think that's the case. I think it's more likely they tag the memory somehow, and they remember that that one's been emptied. We know it's based on local landmarks, because if you let them cache, and while they're out of the room, you remove the landmarks in the room, the posters on the wall and the rocks and stuff you put in the room, or you scramble them, they can't find the seeds. Okay, they clearly can't smell them. We know they can solve geometric problems like halfway in between and isosceles triangles. Julie Jones was an undergraduate and master's student in my lab here at UNL, and I published a series of papers on that. So we've studied a lot of things in Clark's Nutcrackers. I can easily talk for 2 hours just about our work on Clark's Nutcrackers, and doesn't bore me, but it often bores the audience, so I won't do that. But there is one more question I do want to address because I think it's a fundamentally important question, and that is, what I put on the slide here is how general is it? The it is the relationship between natural selection and cognition. How general are the effects of natural selection on cognition? And what I mean by that specifically is Nutcrackers are really good at a lot of spatial problems. Sorry. Nutcrackers are really good at a lot of spatial problems, and the reason we think they're good is the evolutionary hypothesis is over evolutionary time, nutcrackers who could remember where their seeds are better than other nutcrackers, found more food, fed it to their young, had more offspring, and therefore this ability was selected by natural selection. Now, there's nothing in that hypothesis that says that the selection could not have been specific to finding caches. Maybe what happens is when the bird makes a cache, something happens and it imprints that site on his mind. He takes a snapshot of the site. We know that he doesn't do that, actually. We did an experiment on it, but for example. And so the nutcracker is better, has a spatial memory ability that it can apply to cache sites, but not to anything else. On the other hand, it's also possible that selection worked in a way that it affected the nutcracker's ability to remember anything and everything. Okay, that very general effect. So how general, and there are in-between answers possible. It's not just a specific or general, it would be a continuum. And I'm sure the answer is different for different cases. And I do know of a couple of cases in the animal literature where it appears to be quite specific. Song learning, I think, is one good example of that in many species. Well, how do we address that question with nutcrackers? Nutcrackers are a really good case because we have a good idea what the evolutionary history is and what the selective advantage is and so on. Well, the answer is actually pretty obvious. Take nutcrackers and test them against the other species in tests of memory that don't involve recovering their caches and see if they do better. Okay? So we did that. We did a series of experiments on that, and they all came out, all but one, quite consistently. Okay? And I'm going to show you the most dramatic one. It's the most dramatic because it's the most different from cache recovery and because the results are the most dramatic. Okay? And this was Deb Olson's idea for her PhD work in my lab at UMass. And then as a postdoc in my lab, she moved to Nebraska with us as a postdoc. And what she did is she decided to take a classic psychological technique called matching a sample and see how nutcrackers would do remembering spatial locations in a matching a sample test. So you take a nutcracker and you stick it in an operant box, and there are two spots that you use on the screen, and the bird goes in for 36 trials a day, and on each trial starts with you pick one of those spots at random and you light it up with a red circle. The bird pets it 3 times, it goes out, and obviously you've trained the bird to do this. It goes out, there's a retention interval, then 2 spots come on, one in the same place, one in the other place, and the bird has to remember which one he pecked, and in this experiment pecked the other one. It doesn't actually matter what the rule is. As long as you have a consistent rule, you get the same results. Okay? And then what you do to test the limits of the bird's memory is you take the length of that retention interval and you start off a very low level and you increase it by a few tenths of a second each day if and only if the bird's performance was above 75% correct the previous day. It's called titration if there are chemists in the room. Okay? And because it's similar to titration in chemistry. And so what you do is then you draw a plot that shows the retention interval. Because it's going to keep going up as long as the bird's performance is better than 75%. Chance is 50%, so 75% is pretty good. Okay? Now, I have to tell you, when Deb brought this proposal to me for a dissertation, I advised her against doing it because I didn't think it would work. I remember fairly clearly what I said, probably not exactly these words, but pretty darn close. If you take a nutcracker and stuff it in a pigeon box, it's going to act like a pigeon. And if it does, it's not going to be a very exciting dissertation. Okay? And she said, no, I really think it'll do it. And I said, well, if you're really sure, okay, that you're willing to take the chance, I'm certainly not going to tell you can't do it. It's your dissertation. I might tell you, by the way, I wanted to do learning set in birds in Harry Hollow's lab, and I wasn't allowed to do it because they didn't think it would work. So I'd be darned if I was going to tell one of my students they couldn't do something, as long as they knew what the risk was. Okay. And so she did it. And it led to one of the most satisfying moments in my career and hers, I think, when we got to present the data at a scientific meeting, because when we showed the slide I'm about to show you to a room full of experimental psychologists, most of whom had studied pigeons in this kind of task, and where 5 seconds is considered really, really good performance, this is what we got for 4 species: nutcrackers, pinyon jays, scrub jays, and another low-altitude jay, the Mexican jay. It was really spectacular. It was really spectacular. And it's really better than this curve shows. We just stopped at that point because the point was made, okay? But we continued to run some of the nutcrackers, and some of them got up to 90 or 100 seconds, and they were still doing just fine, okay? And we stopped because it was just taking too long to run them. I don't know what the limit would have been. And the thing that's hard for people to appreciate, I think, about this is that this really is remarkable performance, because it's a situation where the problem isn't remembering where you pecked. The problem is remembering where you pecked on this trial, and not the last trial or the trial before that or the trial before that. It's called interference. It's like the difference between remembering where you parked your car versus where did you park it today. Okay? I think we've all experienced that. If you park in the same lot all the time, sometimes you come out and you go to the wrong spot. This is really, and it's so different from caching that we take it as impressive evidence that natural selection had a general effect on the nutcracker's ability to remember things spatial. When we did a similar test where they had to remember color, they were no better than other birds. They were no longer the best species. And so the conclusion is that selection was general but not completely general, and it seems to have affected their spatial abilities but not their non-spatial abilities. Okay. I'm now going to switch to the last third. I say I'm running a little late, but I'll try to go through it quickly, but not too quickly to be understood. And now I want to switch to another hypothesis about the natural setting in which cognition could be important, and this is the social complexity hypothesis, sometimes called the Machiavellian hypothesis. In a very important paper published by Alison Jolly, a very well-known primatologist, in 1966, she suggested that the intellectual demands of social living of a particular sort might provide the milieu, the context, in which there was heavy selection for intelligence among primates. And this context was living in a large, stable group with stable group membership that lasted a long time. That's what we mean by large and stable, right? And complex, where individuals recognize each other as individuals and react differently to different individuals, and where there are many relationships potentially to keep track of. So you might need to know who you're related to, you might need to know who you're mated to, you might need to know who you're dominant to and who you're subordinate to, you might need to know and remember who cooperated with you yesterday and who didn't cooperate with you yesterday, and so on. And her point was this really could put high stress on high selection pressure on certain kinds of social intelligence. And there's even some very suggestive data from Joan Silk and Silk's group that was published in Science a couple of years ago showing that baboon mothers with higher social skills raise more young to reproductive age than ones with lower social skills. It doesn't actually show cognition was involved, but I'll bet it was. Okay. Anyway, the thing is, we realized, reading this literature, that pinyon jays, who we were using for other reasons, and who Russ Bald had been studying in the field for many years, and we knew an awful lot about the social life of the pinyon jay, are perfect animals to test this hypothesis. They live in just the kind of groups I described. Large, up to 300 or 400 individuals, stable. Animals join, they either stay in their natal group or switch groups when they're 1 year old and then spend the rest of their lives in that group. They mate for life. They have kinship relationships. They have helping at the nest in some families, but not others. They have dominance relationships. And they have close relatives who aren't so social, as I'll get to in a few minutes. And so, wow, so we decided to start looking at social cognition. And in this case, the William Allen Bond and I have been leading this project, and I want to take this opportunity to say that we have just about a perfect collaboration. It's been a total pleasure ever since we both moved to Lincoln to be able to work together. And we've worked together on a number of things, some of which I don't have time to talk about today. And, anyway. But Alan and Russ Balda and Guillermo Pazimino, who was a postdoc in the lab at the time, and a host of Pinyon jays, we decided the particular type of social cognition we wanted to work on is a type of inference called transitive inference. And so transitive inference, a transitive system, is a system in which if A is greater than B and B is greater than C, then you know without measuring it that A is greater than C. Systems that have that characteristic are called transitive, okay? Many systems are transitive. Body weight is obviously transitive, okay? Size is transitive. Most quantitative things are transitive. Some things are not transitive. Just ask any football coach, okay? The fact that, you know, the fact that we beat somebody and they beat somebody doesn't mean we're then going to turn around and beat the team they beat, okay? Unfortunately, or fortunately for us perhaps, in the case of Oklahoma, if it was a completely transitive system, there'd be no need to play the game. So I sincerely hope it's not a transitive system. Anyway, one thing that is very commonly transitive in animals is dominance. And so in most dominance hierarchies, and certainly this is the case with we knew this from the field, but we also looked at it empirically in the lab, that if bird A is dominant to bird B and bird B is dominant to bird C, then you know logically that bird A is dominant to bird C. Okay. The question is, if bird A is subordinate to B, he's interacted with B, and he's been dominated by B, and he sees A dominate B, does he have the ability to infer that when he meets A, he should act subordinate without ever having met A before? If he can, under the right conditions, it's transitive inference. Okay? It shocked us. I don't think that's an unfair word to use. It certainly surprised us that when we went to the literature, we could not find a single well-controlled study demonstrating transitive inference in social settings in any animal. Okay? Not one. There were 3 studies, I think it was, that claimed to show it, and they all lacked the appropriate controls. And I'll explain that when I describe what we did, because I don't think what we did lacked the appropriate controls. And nobody's pointed it out to us yet, so I don't think we did. Anyway, but the first thing we needed was a situation in which to look at dominance in Pinyon jays, and so I'll show you one more clip. Okay, so here's Pinyon jays. This is the apparatus, and so there's 3 chambers, and a bird goes in each side. There's a peanut in this cup in the middle, and there's a clear plastic door. These birds know each other. They've been paired with each other a number of times before, and this bird is dominant. Okay. Now I'm going to show you the thing. So what happens is the plastic. Oops, no, I got to do this for this one. The plastic doors are down so they can see who their opponent is. Oh, you see, dominant bird goes and gets the peanut. The other bird looks away. He's interested in the peanut, but he doesn't really try to get it away. Now a little later in the session, okay, they've switched positions, and now the dominant bird is here. And look, the bird, he look away, he fluffs his feathers, he pecks at his feet, okay? It's really quite easy to tell. Once you've watched this for a while, and you've scored the behaviors for a while, you can tell pretty quickly who's subordinate, who's dominant, and presumably so can they, okay? So that was our situation. Here's how we did the experiment, okay? We set up 4 separate groups of male Pinyon jays. I'm just going to talk about 2 to show you the logic, okay? So 2 of the groups had 6 members each. In this group, we'll call the birds A through F, and A was dominant to B, B was dominant to C, etc. And similarly for 1 through 6. I'm just doing that for convenience of talking about it, okay? And we know that those dominance hierarchies were what they are because all possible pairs within each group were tested at least 6 times, okay? And they settled down and they were quite predictable, okay? And the birds, if the birds can do what we're testing, if they can do what we thought they could do, they should know it too, right? Because they've got a lot of experience with all of the members of the group. They live in individual cages. And they only interact in this apparatus, okay? So all that work, and that's 15 pairs in each of these groups, each tested 6 or more times. That's 180 tests just for these 2 groups. It took 2 people working on this almost full time for almost 2 years to get the whole data set. Okay, and without the support of the University of Nebraska as well as the NIH, it couldn't have gotten done. So we're really appreciative of that. Okay, that just set up to do the experiment. The next step was to then take selected cross-group pairs, and we did many of them. And I'm just going to talk about one to show you the logic. We took B and 2, and we tested them. There's no way to know who's going to be dominant. You have to test them and let them figure it out themselves. And let's say that it turns out that B is dominant to 2. Okay? If B is dominant to 2, then 3 is going to be the subject. If B is dominant to 2, 3 is going to be the subject. If 2 had been dominant to B, C would have been the subject. But let's just say B was dominant to 2, okay? Then what happens? Then if 3 is put in the experimental condition, he already knows that he's subordinate to 2 because he's had all those experiences with 2. Now he gets to watch A beat B and B beat 2. 2 is from 3's group, okay? So he's seen B win and lose an equal number of times because sometimes just seeing a bird win will affect the way another bird reacts. So we try to balance it out, winning and losing. And so now, when 3 encounters B, he knows that B beats 2, and he knows that 2 beats him. If he can do transitive inference, he should be subordinate to B when he first encounters B for the very first time. Okay? The trouble is, there's a host of ways you could get exactly that result without transitive inference. That's why scientists like to use controls. Okay? And so there's a critical control. For example, supposing that there's some physical characteristic that determines the outcome of the dominance hierarchy. And we know it's nothing obvious in like body size in pinyon jays, but suppose it's the distance between their eyes or the length of their middle toe. I mean, who knows, okay? You got to take into account everything possible. Then you would get exactly this result even if 3 never saw this. Okay? And so it wouldn't be transitive inference, but it would look like transitive inference, so you need the right control. And there are actually several things to control for. And that was the hard part, okay? And this is when we found, I think we came up with it. So 3 could also have been assigned to a control condition, in which case he would have seen A beat B and B beat C. Now remember, 3 only got one or the other, okay? I'm just using this for example. He would have got A beating B and B beating C, so he sees B win and lose an equal number of times, just like in the other condition. But these are all strangers. There's no possible way that he can index this observation to his own status. And so he should not act subordinate when he first encounters B. But if it's a physical characteristic or something like that, it should on the average work out the same as the experimental condition. Okay? Well, I wouldn't have gone to all this trouble of explaining all this to you if there was no difference. There was actually quite a dramatic effect. Okay? And the experimental observers acted more subordinate. This is a composite index of subordinate behaviors. And right in the first minute of the first encounter, bang, they act subordinate. And it's quite dramatic because pinyon jays usually don't act subordinate when they first meet another male pinyon jay. Now, see where I am, yeah. So I've got one more thing to go. It's going to take about 5 minutes. Is it all right if I just go? I'm right at 45 minutes. Is it all right if I just go? Okay. So what we wanted to do next is we wanted to do a comparative study of the pinyon jay with a less social animal, and it happens to be a species we already had in the lab, the scrub jay, who are much less social. They're territorial, they live in very small family groups compared to pinyon jays, and they're temporary groups. They're not stable for a long time. And so the social complexity hypothesis would predict that these birds should be less capable of transitive inference than these guys, okay? The trouble is we can't do the study I just described to you, probably can't do the study with scrub jays, because they live in these small groups. They probably don't give us stable, long, stable dominance hierarchies. If we get the funding we've asked for, we'll try, but we don't expect to be successful. But it'll be worth a try. So what we decided to do, for 2 reasons, one the one I've given you and the other I'll give you in a minute, was to use an operant approach to transitive inference. It had been very successful for us in spatial cognition, so we thought, why not try it in social cognition? Because there is an operant method that's been applied to the question of transitive inference in animals, and it involves putting birds in skinner boxes again, okay? And so what Alan and Russ and I did is we taught pinyon jays and scrub jays what we call an arbitrary implicitly transitive list. So it was a set of 7 stimuli with the relationship that A was better than B, B was better than C, and so on, because when A and B were presented together, the bird got rewarded for choosing A but not B. When B and C were presented together, got rewarded for choosing B, but not C. When C and D were presented together, got rewarded for choosing C, but not D, and so on. And so you could solve this as a series of 6 separate problems, each one of which has nothing to do with any of the others. Just learn when I see A, B, pick A, when I see B, C, pick B, and so on. Or you could conceivably learn this and pick up the implicit order. Okay? So the way you do these experiments, I'll show you the way that the trials actually look like to the bird, more or less. So if this is the sequence, we scramble, each bird gets a different set of colors in the different positions as a control kind of thing. So they go in for 36 trials a day. On one trial, they might see yellow versus purple. This is not on the screen. This is for us, for mnemonic for us. The birds don't get the mnemonic. And then yellow is going to be rewarded because it's higher in the list than purple. On the next trial, it might get amber and purple. Now purple's correct because when it's with amber, it's the higher one. And the next one, it might get blue and green. And that blue's correct because this is the higher one. And you train the animal until it's doing well on all of these pairs simultaneously. Okay? And then you give him tests with novel pairs he's never seen before, like green and gold, I guess it is. Okay? So he's never seen green and gold, and if he's learned this as a separate, a completely independent set of pairs, he should have no idea what to do. Okay? But if he's learned the implicit sequence, he should choose green. Now, there's a relatively large literature on this in pigeons and rats and some work in primates as well. And there are a number of different models, and there are ways to predict this performance in terms of choice that don't involve, the critical question to us is, does the animal's representation of this set of stimuli have a transitive component to it, the way the animal mentally represents the stimuli? I use the word mentally. Alan's going to give me a hard time tomorrow. Anyway, but there's one prediction that only the transitive representation model makes, and that is the further down the list you go when you ask the question, the longer it should take if the animal's counting down from the top of the list. So think about it. So here's a list, okay? If you've learned it as a list, A, B, C, D, E, F, G, and the response to a pair requires you've got a transitive representation, you've got it like a list in your head, and you have to count down. So if you're asked for B versus C, you have to go A, B, B, C. That should take less time than if you're asked for E, F, in which case you have to go A, B, B, C, C, D, D, E, E, F, right? So our prediction was that as we went down the list, the amount of time it took Pinyon jays to respond would get longer, but the amount of time it took scrub jays to respond would not. Red is Pinyon jays. This is, as you go down the list, this is with the training pairs. This is with the test pairs, some of the test pairs. And you can see there's very little consistent pattern with scrub jays, but it appears to take about 1/4 of a second with the training pairs that they were familiar with to go down one step down the list from BC to CD, from CD to DE, from DE to EF. And it takes about twice as long with novel pairs. Okay. So it's very strong confirmation of the prediction. Now, we don't view these data as complete. We're not satisfied that we've got the right answer. It's not enough data. We need to do some different studies. And we've already done a couple of them. We're writing them up now, working with a postdoc in our lab, Cindy Way. We've worked on a thing called list linking, where you teach them 2 transitive lists, A, B, C, D, and 1, 2, 3, 4, and then you just treat them D and 1 and see if that makes them be able to transfer. And Pinyon jays do it quite well. Really quite well. And so we're doing more, and we hope to have a more complete picture in a couple of years. But again, this work takes time. So, in conclusion then, no, bird brain's not an insult, at least not if you're a corvid or a parrot. But I wouldn't want to be called a pigeon brain or a chicken brain. I hope also that I've shown you that a Darwinian approach to the study of cognition can pay off, is an important approach. And it really can lead to interesting, exciting discoveries and stands in contrast to the more anthropocentric approach, which has its own pluses and minuses. And I hope also that I've shown you 2 instances where the relationship between natural selection and cognition is general, where natural selection has probably affected cognition, almost certainly in the case of spatial cognition, less certain in the case of social cognition, in ways that are general. And it shows us that what we think about human intelligence being applicable not just in one setting but in multiple settings really could be the result of natural selection. It's also another indication to me of how important it is that when we think of our species, we have to understand our species is one of many on the planet, and it's the result of the same, for biologists, it's the result of the same forces, force is not quite the right word, the same factors operating as has affected every other species on the planet for millions of years. Thank you very much.
Thank you all for a very nice talk. One preliminary item before we move on to the question and answer period. There's a reminder that there's the reception afterwards across the hall. If you have a question, we have a microphone here in the center, in the aisle there. Please come up and ask your question from the microphone so that everyone can hear your question.
Or if you prefer, shout it out and I'll repeat it.
How big are the nuthatch caches? How many seeds in a cache?
Nutcrackers, they put between 3 and 8 seeds typically in a cache. In a year of really good pine cone crop, 25 to 30,000 seeds are cached, so roughly 5 to 7,000 caches. That's actually picayune compared to the Eurasian nutcracker, close relative. The pine seeds of the Eurasian nutcracker are quite a bit smaller where they live in Siberian places, and the winters are more severe, and they're estimated to cache 75 to 80,000 seeds a year. In nutcrackers or pinyon jays?
In both.
In nutcrackers, whatever. In the social cognition, in the dominance hierarchy, all male, because males and females have separate hierarchies in pinyon jays, and the male hierarchies appear to be a little stronger, easier to detect. I don't know if they're actually any stronger, but they're easier to detect and measure. They're probably equally strong to the pinyon jays. In the operant work, they're mixed males and females.
We had a question over here and a question over there. Maybe we'll go first.
How much variation did you find in your test groups? Like all opinion jays, how many did you have and how much capability did they differ in? And how did you decide which ones to use?
The question was how many birds did we have in each condition in our experiments, and was there much variation, and how did we decide which birds to use? Well, the answer depends on the studies. In the operant studies, we generally try to use groups of 5 or 6, and we use all the birds that learn to peck. Okay, and that's most of them. But okay, the truth is, some birds just you have trouble training them to peck. It's probably the trainer, not the bird. But and it's easier to start with 7 birds and then take the 6 that learn than to start with 6 and keep trying to get that last 6th one. The funny thing is, a lot of times they're the last one, the one that's the hardest to train, is usually just does fine once they realize, oh, that's what you wanted me to do, In terms of variation, again, it depends. It's very hard to know in the dominance hierarchy work, for example, because the natural dominance hierarchy, because each bird was only tested 1 or 2 times, so you can't really tell from one bird. In the nutcracker cache recovery work, we get about, I'd say, 1 bird out of 20 who just doesn't do it, in the nutcrackers. But I don't believe that's because they can't find their caches. I believe that's because they don't habituate to the laboratory setting very well. And I don't know how much individual variation, I don't remember how much there was in the operant transitive inference work. I'm sure there was some. Some birds are certainly better at it than others. The question is that in the transitive inference work, the birds drew an inference by watching other birds interact. What about during caching? Well, a number of people, including Nikki Clayton's lab and Christine Mueller in our lab, have been looking at the effects of observers. So when birds cache in the presence of other birds, do they respond in ways that make it harder for the other bird to know where they've cached, and do things like false cache, we call it. They look like they dug and it looks like they put seeds in, but you go and you look and there are no seeds there. Okay? Or, Clayton, if I remember right, he's done a study, Clayton and his students have done a study where they put up a barrier, so there was an observer in a cage and it could see part of the room but another part, and the scrub jays either learned to or initially went behind the barrier where they couldn't be seen. Am I right about that? Someone from my lab, am I right about that? My memory right? Basically, more or less? Anyway, so there's a lot of interest in that kind of work, actually. And the question of pilferage is really an interesting question that people are very interested in. It's a good question. Prem?
Al, when you first published your work, talk a little bit about how hard was it to convince others and what you went through, and did you actually have some fights, and especially getting funding, where there's some game playing going on, just to try to get a feel for what sort of environment that you worked in.
Yeah, Prem asked me to talk a little bit about the experience of publishing work we thought was really good, but ran in the face of accepted belief and not having it accepted. And I'm trying to think of a nice way to put this. I was primarily motivated, and I'm still primarily motivated, because I really want to know the answer to the questions. Okay, and I believe in doing really the best research I can, the best controlled research I can, because I want to be able to have confidence in the answers to the questions that I come up. When I come up with the answers, I'm pretty critical of experimental design, as anyone who's worked with me knows, including my own. Okay, and including those of my friends and some of my former friends. But I guess my attitude was, a famous case when I did that Blue Jay work, there was a guy at Penn State named Mike Warren, that graph I took was from a review paper he wrote. The funny thing is I wanted to go to Penn State and be his PhD student. He turned me down, and I'm glad he did. But anyway, I sent him the reprints and met him at meetings and showed him the work and pointed out to him that it really invalidated his conclusions. And he told me, yeah, it's nice work, and I've read it, and blah, blah. And then he wrote another review article, never mentioned it. And you know what? I decided, screw him. I just went ahead and pursued the learning set problem until I thought we'd done everything we needed to do, then moved on to the prey detection work, what I didn't talk about today, and other work on nectar feeding birds that I thought was really interesting. And I probably would have been better advised to write some aggressive review articles, but I didn't. I was more interested in getting more data than I was in fighting about it. Now, the funding question is an interesting one. I got funded. I remember my first grant to do Learning Set and Blue Jays. One reviewer said, it got funded. One reviewer said, well, if this work really needs to be done, this is the right way to do it. But I got funded, so I wasn't complaining. But then when I got to the point where I wanted to do the search image work, the work on the prey detection, where I was combining some very sophisticated operant work with some detailed natural history about the evolution of crypticity in moths, the first grant got turned down, and it got very good reviews. So I called the program director, who I knew quite well because it funded my previous work and so on. And I said, Fred, I don't understand, why'd you turn this down? The reviews were good. He said, well, Al, the problem is when I read the reviews carefully, I saw that all the psychologists liked the biology, and all of the biologists liked the psychology. And he said, and I thought that might be something, that might be a sign of a problem. Well, of course, I'd written all of the biology so the psychologists would understand it, and I'd written all the psychology for the biologists would understand it, and I guess that was a mistake, okay? So I rewrote the grant and realized that I just had to show that actually I'm sophisticated about both sides of the equation here, guys and gals, because there's men and women on those panels. And then it got funded. And, but there were times, we've had a lot of trouble at times, particularly with the NIH, because we're not working with a standard model system. And Alan and I submitted a proposal not that long ago. It's the R21 that we just resubmitted and got a very good score on. The first time we submitted it got turned down flat. And the reason was because several panel members, and they actually said this in their review, and I'm quoting almost exactly because it's burned in my memory, avians can't solve such complicated problems. Avians can't solve such, some idiot psychologist wrote that. I'm quite sure of the psychologist part because I looked to see who's on the panel. But someone who had not read the literature, because in the 3 or 4 years preceding the submission of that article, there had been 6 substantial review papers, none of them authored by us, by other people, in addition to papers we'd published, pointing out just how good Corvids were at complicated intellectual problems, and just how sophisticated the neural apparatus of Corvids was. And they were in obscure journals, I admit, Nature, Science, Proceedings of the Royal Society. I'm not kidding. I'm not making this up. And Alan and I were really beside ourselves. But we revised the proposal, and we put in a section that was very polite. We wrote a draft that we ripped up and threw away. Actually, I've saved it somewhere because it's juicy. And we said, look, you need to go look at the stuff. Actually, there's been a big change in opinion about this and so on. And I might say that some of those papers that I'm talking about by Emery and Clayton and others cited the learning set work that I'd done in the 60s and 70s, and finally getting cited. So if you live long enough, almost anything might happen. And we've got a very good priority score, and it looks like it's going to get. We're just waiting for the Congress to pass a budget, hopefully, and hopefully we'll be funded. So you can. It has definitely been difficult to be at the border between two disciplines, and what's been more of a problem though has been to be flying in the face of what a lot of people think is true and they're wrong about it. But we complain a lot about the old boy network and how the connections determine success on grants and so on. But I have to tell you, I've collaborated and worked with a lot of people from Australia, New Zealand, Germany, England, and they all envy the United States system of grant proposals and grant evaluation because it's a lot more open than theirs. And I have to say, ours isn't perfect, but if you regard what the reviewers write as communication, and then you respond to what they write in an articulate and sensible manner, you can overcome it. I'm sure that a lot of good work doesn't get done because of these things, but a lot of good work can get done. But you've just got to, an old friend of mine said, communication, it's all about communication. So, I'm not in any position to complain about support. I've been supported by either NSF or NIH since 1971. So I think with one lapse of a little while. So I can't complain about the system, and I won't. So, Svacha.
Now, isn't that part of the history of science that every time you propose a new idea, that challenges the paradigm, the way that you perhaps people look at it, is to have trouble at first? Would you agree?
Could you all hear Svacha? Yeah, I'd agree. Sometimes, the philosophers say, it'd be interesting to discuss the writings of people like, the Nature of Scientific, Thomas Kuhn and Imre Lakatos and others have written about this kind of thing. But this isn't the place, probably. Yes.
How is this cognition stored in the brain? A series of visual images. Could you put electrodes on these birds and try to get some idea? I guess you can do it with primates.
Yeah, the question is, how is information stored in the brain? If I knew the answer to that, I wouldn't tell anybody in the room. I'd run home and publish it. But we're really learning a lot, and with the functional MRI and other techniques are adding to our knowledge. Unfortunately, those techniques have not yet got to the point where we can do it with birds. One of our problems is a lot of those techniques are destructive of the animal that's the subject. And we can't, it just takes, you can't take 2.5 years to train an animal to do something just to kill it, you know, and you can't get the numbers. I mean, I have trouble with it ethically, but I have more trouble with it, or just as much trouble with it practically. But I think it's coming. It's certainly coming. There are model systems, including work with Drosophila and simpler systems and looking at genetics of learning. I think the next 20 years going to be pretty darn exciting. I hope to be around for most of them anyway.
Maybe one more question?
Sure, Monica. The question is to talk about the geometry, but it's a short question. So there was a great debate in the literature about whether or not animals could learn the general principle, geometric principle, particularly something like relatively simple, like halfway. Okay, and I'd always thought Nutcrackers might be able to do that, and it was kind of a nice project because if they couldn't do it, I thought they probably couldn't, but they might be able to. And Julie Jones, who is one of those cases that UNL has so many of athletes who are also wonderful students. She was a diver on our swim team. Started working in our lab, and we hired her as an assistant. And I noticed that she was doing these very. I'm going to tell this in a little length because it's a good story, I think, for students to hear. And I noticed that whenever we had some very complicated spreadsheets that kept track of the data. And whenever she went and worked on them and brought them in for me to look at, they were always correct. I never got them always correct. Okay. So I started talking with her and asked her if she was interested in maybe, starting a project. And she said, yeah. And I said, well, this was before you care. But we had equivalent things. And so I said, well, here's a couple things that are laying around. Would you be interested in one? She got interested in this geometry one. So we worked out this technique. And it was perfect for summer because the answer would either be yes or no in the summer if they could do it. And what we did is we put the birds in the room 4 times a day. And there was always a seed buried. Well, sorry, there was always this one seed in the room. And it was placed halfway between 2 landmarks. And those landmarks moved around the room. They were always east and west of each other, but otherwise they moved around the room. And the distance between the landmarks varied in 20-centimeter steps. So sometimes it was 20, I don't remember the exact numbers, but I think it was 30 centimeters, 50 centimeters, 70 centimeters, 90 centimeters, 110 centimeters. Okay? And the first time they did this, the seed is sitting on the surface. Okay? And so at first, the seed's on the surface 3 times a day, and it's buried once a day. And then as they learn it, you start burying it all the time. And then once they've really learned it, you start testing them with no seed every once in a while to make sure they're not smelling the seed or something like that. And then and they learned it. The birds learned it in like 2 weeks. I couldn't believe it. I wasn't totally shocked, but I really was shocked at how fast they learned it. God damn. Sorry. Excuse me. I mean, when some experiments, you pretty much know how you think they're going to come out, and then you often get surprised. And some you really don't know. And when you get such a clear answer, I mean, it's really cool. It's one of the big rewards of doing this business. Anyway, so then you test them with novel distances they haven't seen before, and it made no difference. You could extrapolate, you could go to longer distances than I've never seen before, shorter distances than I've never seen before, in between distances that I've never seen before, and they did it exceptionally accurately. We then did things like we gave them 2 landmarks, and the seed was buried at the 3rd point of the triangle of which those 2 landmarks describe one side, a isosceles triangle, and they solved that just as fast as they solved halfway. Okay. So that's the geometry work. We published, I don't know, 6 papers on it. You did though. I'm glad that you reminded me though. Now, answer the last question. There are 2 things I neglected to say at the end of the talk. Okay, the first thing is that I've been very fortunate in my career to have many collaborators, and I probably left a few off here, and I apologize for that. But another of the pleasures of this work is getting to work with a lot of really great people. Okay, and getting to work on exciting things that you're all mutually excited about. And I did not accomplish the things that were described today by myself. Okay, I accomplished it all in collaboration with other people. And the other thing is that I am somewhat obsessed with what I study, as many of us are, and that really creates painful situations for one's family. and I owe a great deal to my family, and I'm pleased that to say, and I'm going to ask them to stand up, that my wife of almost 50 years, Ellen, is here. If you'd stand up, Ellen, please. And then, of course, I have a lot to thank my parents for, because they encouraged me to follow my interests and do something that really interested me. And I think when I was a teenager, they despaired that anything like that would ever come along. And my father, unfortunately, has passed away, but I'm pleased that my mother is here today, and if she wouldn't mind standing up. Thank you.